Is now starting. All attendees are in listen-only mode.
Hello, everyone. My name is John DeBella, and on behalf of Simulations Plus, I welcome you to today's webinar, where we will describe and demonstrate the new features in ADMET Predictor version 9, which have been implemented in ways to help streamline your drug design activities. David Miller, our Director of ADMET Chem Informatics, will be speaking today. Joining David as panelists are our esteemed Senior Research Fellows, Bob Clark and Marv Waldman. An opportunity to ask questions will take place at the end of David's presentation. You may either type your question using the questions pane on your control panel, or you may ask your question directly using the hand-raising icon. This webinar is being recorded for playback at the online resource center on our website, www.simulations-plus.com. Also, upon request, the presentation slides can be made available and shared with colleagues. It is now my pleasure to turn the presentation over to David. David?
Thank you very much, John. So as John said, my name is David Miller. I am, among other things, the lead software engineer on ADMET Predictor. I want to say in advance that I just spent 10 days in Asia talking about ADMET Predictor with our customers, and I came back with a gigantic cold. So I apologize. You may hear me coughing a bit, drinking some water, but we'll see if we can get through this. Let's see. I want to focus on the new features in ADMET Predictor 9.0. This version is scheduled to be released in the coming days. We are just wrapping up some final testing. I'm going to break the presentation up into a slide portion and a demonstration portion. For the slides, because there may be some listeners who are not familiar with ADMET Predictor, I would like to spend a little bit of time giving a little overview of what ADMET Predictor is, and then I'll start discussing what is actually new in the latest version. Okay, so what is ADMET Predictor? ADMET Predictor is primarily used to predict properties from chemical structure. At the moment, we have something like 140 ADMET properties that we predict. These include physical chemical properties, metabolism, and toxicities, as well as a number of scores that we call risks. One example shown here is the overall ADMET risk. These are scores to help identify potential problems with a compound becoming an orally bioavailable drug. So you can think of it kind of like Lipinski's rule of 5, but encompassing many, many more properties. Recently, in ADMET Predictor version 8.5, we introduced what we call the high-throughput pharmacokinetic simulation module. So that is used to predict fraction absorbed and fraction bioavailable, as well as other properties using a virtual human or rat simulation. So this feature is similar to our GastroPlus software, and we have made a number of changes to this module for version 9, and I'll be discussing those. The ADMET Modeler module is used to let you build your own QSAR models or QSPR models using your data, but using our molecular and atomic descriptors. This is the module that we use as well to build our commercial models. We've made a number of improvements here as well, which I'll be discussing. The MedChem module is used more by chemistry groups in early drug discovery. It has a number of features for things like prioritizing lead series, looking at SAR trends, and designing novel compounds. Okay, ordinarily, we would spend quite a bit of time talking about the predicted properties in ADMET Predictor. In the interest of time, I will give just a very brief overview for any listeners who may be new to the software. One of the modules, so there are several modules, is the physical, chemical, and biopharmaceutical module. So that includes things like pKa prediction, log P, log D at a user-specified pH, several solubilities, both aqueous solubility and in simulated gastrointestinal fluids. There are permeability predictions, including human intestinal, blood-brain barrier, and others. There are transporter models, including PGP, and several PK property predictions, things like protein binding, volume of distribution, etc. The metabolism module focuses primarily on cytochrome P450 and UGT enzymes for several isozymes, I think 9 of both CYP and UGT. There are simple substrate, non-substrate models. So is this compound predicted to be a substrate or not a substrate for 9 different isoforms of the CYPs and the UGTs. For the same 9 CYP isoforms, there are site of metabolism models. So where on the compound is the phase 1 metabolism likely to occur? For the 5 major CYPs, we have kinetic models. So the Michaelis-Menten KMV-max parameters and intrinsic clearance. Those are from recombinant CYP assays. Then we also have an overall predicted liver microsome clearance. And we have a separate model for human and for rat. So that's overall human or rat liver microsome intrinsic clearance. And then finally, we have CYP inhibition models as well. Several different toxicity predictions. This cardiac prediction, cardiac toxicity, including hERG channel blocking, carcinogenicity, including Ames models, liver and endocrine toxicity, skin and respiratory sensitization models, acute toxicity in rodents, and a number of others. And then the HTPK I just briefly mentioned, and I will transition to a few slides that talk about that module in a little bit more detail. Okay, so in ADMET Predictor 8.5, this would have been late last year, we introduced this HTPK module, meant to be essentially a simplified version of our GastroPlus software, more appropriate for early discovery. So we were really focusing on sort of a faster way to get a handle on PK predictions in early discovery. So it's based on the same absorption model called the ACAT model that's used in GastroPlus. I have a slide in a second to show a little more detail about that. Again, you can predict fraction absorbed and fraction bioavailable for a given dose or doses, as well as predicting the dose required to achieve a target plasma concentration at steady state. For the moment, we are supporting both human and rat species with possible plans to introduce other species in the future. And like GastroPlus, you can use predicted properties, which presumably are from ADMET Predictor, or if they are available, you can use experimental values as well. Just a brief look at what is happening in vivo here. So we are simulating the absorption of compounds that are delivered orally, compounds that are able to permeate from the intestine into the enterocytes. That is how we define fraction absorbed. Compound that survives first pass through the liver is used to calculate fraction bioavailable. At that point, we are essentially performing a compartmental simulation. This is not a full PBPK simulation at this point, although that is also something that we are considering as a future development in the software. And then just looking at the model itself, excuse me. So this is a schematic of the GastroPlus Advanced Compartmental Absorption and Transit model. So this is the model in which the gastrointestinal tract is divided into compartments, each with its own unique physiology and transit time and other parameters. And we are, through a set of differential equations, modeling the amount of drug that is undissolved, dissolved, or that has permeated into the enterocytes. Most of the parameters, for this model can be adjusted just as they can with GastroPlus. And then, of course, as I mentioned, we have the compartmental simulation that involves both liver metabolism and renal clearance. Let's see, just a few notes about the differences between ADMET Predictor and GastroPlus. So the ADMET Predictor simulation is not modeling clearance that occurs in the gut. It is not modeling active transport, so it's only passive diffusion. And we are not modeling nonlinear kinetics. So when you specify a clearance value, it is an intrinsic clearance value. As opposed to individual Km and Vmax values. Let's see, this slide was really meant to discuss kind of the motivation for us adding this simulation feature to ADMET Predictor. This is, kind of targeting early discovery scientists who, in our experience, will have a tendency to focus on individual properties, things like solubility and permeability. So we are proposing kind of an alternative mechanism, where instead of optimizing individual properties, you can run a virtual simulation and then focus just on those properties that are predicted to be most important for absorption and bioavailability. So I think maybe for DMPK groups, this maybe is not a big paradigm shift, but for early discovery groups, this is a little different than maybe they've traditionally thought about discovery and optimization. Okay, I'd like to just spend a few slides talking a little bit about how we create our models, and I want to just touch on how we evaluate the accuracy of our models. This is a kind of an involved slide here. I just want to talk about part of it. Several years back, under a grant, we wanted to develop a way to estimate accurate quantum level charges. So under the grant, we spent a great deal of time calculating quantum level partial atomic charges on a large set of drug-like compounds. This was over a period of many months. Just the computer time was many months. We then developed a fast empirical methodology to estimate those partial atomic charges using just 2D structure information. So a very fast estimate. This graph shows the result of that work. So the x-axis is the estimated charge using this empirical approach, and the y-axis are the actual quantum level partial atomic charges. So you see a very close fit here. This shows I think the number of okay, maybe I don't see it here, so I don't know how many charges are here, but there's many thousand, obviously. Using these estimates of the charges then, we've developed a number of proprietary atomic descriptors that we can then use for model building. Some of the descriptors are listed here, something like 150 descriptors per atom. So this slide is showing the predictive performance of some of our models, all of which are based on those descriptors that we've developed. So here I'm showing the PKA model, log P model, and solubility model. If you evaluate or purchase the software, these types of statistics are available for all of the models that are in ADMET Predictor. Not just the statistics, but a description of where the data came from. So you can see the source of the data that we have used to build our models. Recently, there have been a number of third-party comparisons of predictive software. And we make sure to collect those. They all, as far as we're aware, show that the Simulations Plus software does extremely well. I don't have time to go over all of these, and I believe they should all be listed on our website. The ones we're aware of are for log P, solubility, and PKA. I want to draw attention to the most recent challenge, which was called the Sample 6 PKA challenge. We were notified of the results of this one, I believe, in just the month of May. And I think I'll have to have one of the panelists correct me if this is not actually the absolute latest result. But the one that I'm aware of is that the only predictive software to outperform ADMET Predictor was one that was based on a full quantum chemical free energy calculation. I don't know that we have a lot of information about this, but we assume that it must be a very time-consuming calculation. So the website listed here will provide more information about that challenge. But our software in general, in the hands of third-party scientists, has performed very well relative to other predictive software. Okay, so that was kind of the basic introduction. So now I want to describe some of the new features in the latest version of ADMET Predictor. Okay, so we have added some new graphical tools, including ones to show both log D and solubility as a function of pH. This has long been a feature in the GastroPlus software, but up to now it has not been available in ADMET Predictor. We have a set of new models to predict the major clearance mechanism for compounds. I have some slides to go into that in more detail, but this is related to the ECCS classification system. So I'll talk more about this in a minute. We have introduced some new visualization tools to look at how compounds are classified in both the BCS and the DCS classification systems. I have a couple of slides to talk a little bit more about this in a second. We have spent some time improving some of the models. Two in particular are the plasma protein binding model for human, as well as the human liver microsome intrinsic clearance model. I hope I didn't get that wrong. We do have two models. One is rat, one is human. I believe the one we improved was human. And this was through the acquisition of new data. We also improved the confidence estimates in all of our classification models. So the classification models are those that are, for example, yes, no, toxic, non-toxic, or substrate, non-substrate. So we have developed a proprietary methodology to provide a confidence score along with the actual prediction. And to introduce this new methodology of providing confidence estimates, we had to rebuild those models. So in ADMET Predictor 9, all of those models will have improved estimates of confidence. Okay, this is just a graphic showing what the log D profile looks like now. So you can select a single compound, look at the predicted either log D or solubility as a function of pH. You can also show as vertical lines where the pKa, the predicted pKa are for those compounds. So this is sort of a useful companion to the other advanced visualization that we have for looking at pKa. And I think I'll have a chance to show that during the demonstration. This is a graphic from a recent publication from El Katan and Varma at Pfizer. So they have published on this system called the Extended Clearance Classification System, or ECCS, where they are predicting the primary clearance mechanism for compounds based on the physical chemical properties of the compounds. So in their scheme, there are 6 classes that are actually overlapping here. So we encourage you to look at these publications. The scientists were kind enough to share their data with us, which we have used to build a number of different related models, 5 of them. One of the models that we built is meant to closely mimic the Varma-El Katan model. So it's a model that predicts for each compound one of 6 classes, just like the original ECCS system does. The difference, of course, is that instead of using experimental properties, experimental pKa and permeability, it's using our predictions. But it closely reproduces the ECCS, the actual published ECCS classification on that same data set. We have also developed 3 binary models using the same data. So one for metabolism, one for renal, and one for hepatic uptake. So each of these models predict either yes or no for each compound, along with a predicted confidence score. And then finally, using one of the newer features that I'll talk about in a second in ADMET Predictor 9, we developed a single 3-way model. So for a single compound, this model predicts either renal metabolism or hepatic uptake as the primary clearance mechanism. So these are all related models, and for the most part, you know, they usually agree, but there are cases in which they don't always agree, which is not so surprising, since you know a compound may have a mixed, you know, may have more than one predominant route of clearance. And this is showing the interface for looking at BCS or DCS class assignment. This is not really a new prediction. It's using you know, current predictions from ADMET Predictor, but it's allowing you to visualize this in sort of the convenient, you know, 4-quadrant BCS system display. So what you can do is from ADMET Predictor get a prediction of both solubility and permeability. You can enter 1 or more milligram dose values. You can, you know, just actually manually type them in here or load them from the ADMET Predictor spreadsheet. If you have experimental solubilities or permeabilities, you can edit these values dynamically within the window. Let's see, for each compound, then, if there are multiple doses, you actually see multiple points then. So one for each dose value. And then there's features to kind of include this graphic in reports and whatnot. I should just say that we were asked to add this functionality to ADMET Predictor. There are companies for whom this type of analysis is required in development, and so we were asked to add this to help simplify that process for some of our customers. You can dynamically switch between the BCS system and the DCS. I did not put the reference for DCS into the slide here, but this is a system that's related to the BCS, a little bit more skewed toward formulation. So there's something here called the SLAD line, the solubility limited absorbable dose. We have a pretty nice description of this in the user manual for ADMET Predictor. Okay, we've added some new features related to ADMET Modeler. One of them was to simplify the workflow for building what we call delta models. So I have a couple of slides to talk about that. And then we've extended the neural network method of building classification models. So again, these are typically the yes-no type of models. We've extended this to be able to work on data with more than just 2 class labels. So I'll show one slide to explain that. So briefly to talk about delta models, this is something again that we introduced in the previous version of ADMET Predictor 8.5, but we've simplified the workflow for 9.0. So just to review, a global model, by which I mean typically a simulations plus model. So this could be a log P model or a rat liver microsome clearance model. These models may not always perform well on your particular data set. Assay conditions may differ. Compounds may contain somewhat unusual substructures that were absent or that were underrepresented in the data that we used to build our models. Customers, typically use ADMET Modeler to build models on their own data. We call these local models. But these are often restricted to a fairly narrow chemistry space that a company happens to be working in. A delta model uses one of our global models as a starting point, but it incorporates an error model that is internally built on the difference between our global model prediction and the experimental values. And then the delta model, the final delta model, combines the prediction from our global model and the error model that's built as part of building the delta model. So the advantage is that this delta model often can outperform a local model, and it incorporates information from a broader chemistry space. The data that we use to build our models typically comes from quite a broad chemistry space. It may be much broader than the more narrow chemistry space that a company is working in. So that's kind of the background on delta models. What we've done in ADMET Predictor 9 is to make it a lot easier for you to experiment with building delta models. And hopefully there'll be time for me to show a quick example of this after the slides, but basically you can simply select what we call the reference variable. So that's usually a simulations plus model that you want to use as the basis for your delta model. Okay, and then finally with ADMET Modeler, this is just showing how now with neural network classification models, you can use data that has more than 2 classes. So this is taken from the example I referenced previously for major clearance mechanism. So here the data, each compound is assigned as being either hepatic uptake, metabolism, or renal. So this is showing the, what is typically the confusion matrix with just 4 quadrants, and now it has the 9 quadrants. We did have this methodology with our support vector machine method, but until ADMET Predictor 9, it was not available for neural networks. Okay, on the high throughput pharmacokinetics module, We now provide predictions of other PK parameters, Cmax, Tmax, and AUC. We have, for a single compound, you can display concentration versus time curves. I'll show one slide on that. And there is a parameter sensitivity analysis feature. Right now, it is limited to just two parameters, solubility and permeability. So you can look at how each of those parameters is predicted to affect major PK parameters like fraction absorbed and fraction bioavailable. This feature has long been available in GastroPlus, of course. Let's see, this is just an updated slide showing the performance of the HTPK module. This is looking at fraction absorbed. This is on a set of 125 passively absorbed compounds from this data set referenced here. I'm sure we showed this during the 8.5 webinar. We have made some improvements both to the simulation. We have corrected some errors that we discovered in this particular data set. Of course, I mentioned that we have made improvements to the plasma protein binding model as well. So overall, the performance is a little bit better now. So again, this is showing the predicted fraction absorbed from the HTPK module versus the actual observed fraction absorbed for these compounds. We're looking at roughly 90% of the compounds predicted within twofold of the observed value, and roughly 83% predicted within 1.5-fold. We've started looking at whether we can rationalize some of the outliers on the basis of whether they are substrates for PGP or UGT. If you remember, we are predicting, or rather, we are not handling active transport in our simulation, and so a compound, that is a substrate for a transporter would be expected not to be predicted well in our simulation. We have not completed that analysis yet, but it's something that we're looking at. As far as fraction bioavailable, this is a similar performance plot on a published data set from Tashimoto. Here, 62 compounds that are primarily CYP metabolized. So, one of the inputs to the simulation is an intrinsic clearance, for which we are using an ADMET Predictor predicted intrinsic clearance, but it is specific to cytochrome P450s. So these are, this is a subset of this data set that include only the primarily CYP metabolized compounds. Here, the predicted values of fraction bioavailable are, let's see, roughly 81% within twofold and roughly 68% within 1.5-fold. Slightly better than in the 8.5 version. I also briefly just want to mention that we are beginning a couple of collaborations now with companies that should allow us to obtain a much greater amount of pharmacokinetic data, mostly in rat, but probably some in human. And we hope this will allow us to extend these validation sets and hopefully improve the model significantly. This is a screenshot just showing the plasma concentration profiles. This is for a single compound, but you can specify more than one dose. So this is, again, the result of a compartmental simulation in ADMET Predictor. The agreement with the compartmental simulation in GastroPlus is very good quantitatively, not just qualitatively, given the same inputs into GastroPlus. Of course, yeah, you can see the predicted PK parameters here as well, and you can click on the different doses and change the graphic. And then this is showing what the PSA, or the parameter sensitivity analysis feature, looks like. So again, it's limited to solubility and permeability, but you can look at how those two parameters are expected to affect, for example, fraction absorbed. So this is showing a compound that improving the fraction absorbed would require increasing the permeability, whereas changing the solubility for this compound is predicted to have very little effect. Again, this is a feature that is widely used in the GastroPlus software. And then finally, just a few more miscellaneous features. We have been moving much functionality from another piece of software called MedChem Studio, moving that functionality into ADMET Predictor. One of the last features was one to calculate and visualize a set of structure-based descriptors. You could think of them as, I guess, fragments or fingerprints. So these have a wide variety of uses, and I have a couple of slides on that. We've made some small improvements to the display of predicted metabolites. Hopefully I'll have a chance to show that in a second. Modest improvements to the rules for standardizing tautomers, and a number of more minor enhancements and bug fixes. This is showing this feature we call keys. You can think of them as fingerprints. And I'll try to get a chance to show this, but in case I don't have time, this is kind of what the interface looks like. So for a set of compounds, you can calculate a set of fingerprints, which are shown in a spreadsheet as highlighted structural elements here. And then if I advance the slide here, I can show that one of the things you can do with this is look at whether particular structural fragments tend to correlate with a particular property. And this data set happens to be one that has experimental PIC50 values against HIV integrase. So what the histogram is showing is that the particular fragment highlighted in red, the compounds that have that highlighted fragment tend to have a much higher activity distribution. So those compounds are shown in red. The compounds shown in blue here are those that do not have that particular fragment. So the distribution of activity values is skewed to the left. So this is done automatically. You can say, show me the keys that most correlate with, any property that's in the spreadsheet. So you can quickly discover those that are most significant, those fragments that are most significant. There's a few different kinds of fingerprints. We've added the popular extended connectivity fingerprints that are used probably most often through Pipeline Pilot. You can also just define your own fingerprints using substructure queries. Let's see, do I have another slide on this? Oh yeah, I do. The other useful feature of this keys interface is looking at what we call structure alerts. So these can be related to properties like toxicity, assay interference, or reactive metabolites. We worked with one of our customers to develop a set of fragments that are, at least in the literature, reported to be related to reactive metabolites. And so with that set of defined queries, you can load a data set and then get an interface like this, where you can actually see where these particular fragments hit your compounds. You can see how many of your compounds are predicted to have that or that have that particular fragment, and see what the alert is for that fragment, and then do further analysis on it. So this is just another kind of visualization tool that's part of our cheminformatics platform. Okay, excuse me for 1 second here. Okay, sorry about that. Okay, that was the end of the slide portion. So now let me spend a little bit of the remaining time walking you through just a couple of examples with ADMET Predictor itself. So this, I'm actually using the soon-to-be released ADMET Predictor version 9. ADMET Predictor works by opening up data files containing chemical structures and associated data. For example, SD files or SMILES files. I'm going to start by opening up a small set of commercial drugs, about 300 drugs. Okay, so you'll be familiar with the ADMET Predictor interface. It's a a spreadsheet application, a lot of functionality that we won't have time to discuss, just related to manipulating structures, searching, sorting, plotting, etc. I just want to briefly walk through the typical process for predicting properties. It's quite straightforward. I mentioned the different categories of properties that we have: physical, chemical, metabolism, toxicity, and modeling descriptors. So these primarily being used to build your own models via ADMET Modeler. Any models that you've built with ADMET Modeler would be available as well to predict. Software is generally quite fast. This is again about 300 compounds. Following the run, of course, you have something like hundreds of predicted properties, including the overall ADMET risk, or the sort of the ADMET liability score for the compound, pKa values, log Ps, etc. Just as a reminder, some of the advanced visualizations, I can search for a compound. This is a obviously an antibiotic. Can look at the predicted pKa's as numbers, so predicted both to have an acidic pKa and a basic pKa. There's an advanced visualization where you can look at the actual predicted ionization microstates for this compound and show them as a graph that's giving you the predicted population of these microstates as a function of pH. So you can see for this compound, the neutral Zwitter ion, this ionization state, is predicted to be predominant at physiological pH. And then with a new feature of the software, you can look at what is the predicted either log D profile, or in this case, the predicted solubility profile for that compound. So it kind of closely mimics the graph that I just showed for the ionization states as a function of pH. And you can, of course, show what the predicted pKa are, and then map it onto the predicted solubility profile. So the neutral Zwitter ion, which is dominant here, that has the lowest solubility. Just as a quick reminder about one other feature that I'm showing, aripiprazole, the drug, as far as metabolite prediction, or as either metabolites or metabolism prediction goes, we have two kind of nice visualization features. One is called the atomic properties display window, where you can look at either, for example, predicted sites of metabolism for a particular CYP isoform. This is 3A4. You can even show more than one CYP at a time, or actually showing the quantitative predictions of intrinsic clearance for a particular isozyme, again 3A4 here. So these are actually in units of microliter per minute per milligram HLM protein. In addition to that type of prediction, you can also send a compound to our sketch software called MedChem Designer, where in addition to sketching compounds and predicting their properties on the fly, you can also get a sort of a tree of predicted metabolites. So this is the parent structure and then showing you what the predicted metabolites are. Sometimes this is just a simple hydroxylation. Sometimes it is that followed by a predicted spontaneous decomposition to a more stable observed metabolite. Let's see, we show the enzyme predicted to be responsible for the metabolism, the predicted intrinsic clearance, and the predicted yield for each of the metabolites. At this point, you can also, predict the properties of the metabolites, right here within this interface. It's a sketch interface, so you can make changes as well. And I do have to delete that fragment because ADMET Predictor at the moment won't work for a mixture like that. So that was a prediction of a metabolite that involved two individual components. So I deleted one of them, but at this point I can, predict properties of all of these metabolites. And I should mention we do have agency customers who use this feature to predict metabolites of compounds and then use ADMET Predictor to predict the toxicity of those metabolites. So this is a useful feature for that. You can also, because this is a sketch tool, make changes to the compounds and then dynamically predict how that would affect predicted properties. So you could, for example, add a blocking group to a site and then predict the metabolites of that modified structure. So here you get one less metabolite because you've blocked one of the groups. Okay, that's just a very, very fast overview of the kind of the basic property prediction features of Predictor. I want to show just a interesting example that involves the high-throughput PK functionality of Predictor. To do that, I'm going to actually design some analogs and do some predictions on those designed analogs. I'm going to use a feature called R-list explosion, which is going to allow me to basically draw a scaffold, any arbitrary scaffold. And I'm going to pick one that's related to a commercial drug. This could be drawn with our sketch software, or actually with any commercial sketch software, and just paste it in here. There are two R groups, or two variable positions on the scaffold. For each one of them, I can provide a list of substituents that I want to use. I actually have a file where I've enumerated those. I could show them in the sketch tool. There's 4 for R1, just simple substituents, and then I can choose some for R2. Again, I can show them in the sketch tool. These are also quite simple substituents. So given the 4 and the 7 substituents, you can essentially explode those products out. So that should give me 28 virtual structures here. Now, one of these actually is a commercial drug. I'll talk about that in a second. So these are 28 structures. I can, of course, predict properties for those, as I just did. And now I'm going to run the HTPK feature. So I'm going to click this button to launch the dialog for predicting fraction absorbed and fraction bioavailable for these compounds. I think last time, last webinar, we did spend some time going over the options here. For the bioavailability, you do have to provide a clearance. By default, this is an ADMET Predictor prediction of overall HLM intrinsic clearance. Of course, you can specify an experimental value. There's also an option for hepatocyte if you have experimental data for that. Advanced parameters, of course, there's a log P or log D, solubility, permeability, etc. And I should just briefly point out that there are a number of other parameters that can be modified not through this interface, but through a file that's included in the software. But things like, you can, if you have experimental pKa values, you can enter those. Formulation parameters like particle radius can be modified. So let me accept the defaults, except provide a dose value in milligrams, and I'm going to use the dose that is actually appropriate for the one commercial drug that's in this data set. So it's 30 milligrams. If I run that, it's basically going to provide me with predicted fraction absorbed and bioavailability values for all of these compounds, as well as other PK parameters, which are new in ADMET Predictor 9. You can use the histogram to plot what the predicted fraction absorbed values are for these compounds. And they actually range quite a bit, from about 40% up to more than 90%. Down here, I'll show a plot of predicted fraction absorbed as a function of predicted fraction bioavailable. Those match pretty well for these compounds. It's because the predicted rate of metabolism is relatively low for these compounds. Let's see, just to show off briefly some of the other cheminformatics capabilities of the software, let me take you to the Classes tab, which can be used to cluster these compounds. We kind of know everything there is to know about these compounds since we created them virtually, but let me show you what it would look like to cluster these compounds using one of our clustering methods. The blue highlighting indicates the common scaffold for these compounds. So as we know, all the compounds share this common scaffold, the R groups being attached. We can build the R group directly within ADMET Predictor. It shows us just what we expect to see. We can visualize that R table in the traditional MedChem type of R table, where the compounds are in the rows and the two R groups are listed as columns. Or we can view it in a somewhat different way. For example, one in which the R substituents are listed as rows and columns themselves. So this is kind of a matrix view in which the unique R substituents are listed as the R1 substituents are listed as rows and the R2 substituents as columns. The cells indicate the compounds. You can color that by property. So I can color it by predicted fraction absorbed. I can then sort by that predicted property. So the compounds predicted to have higher fraction absorbed are shown in the upper left, those with lower predicted fraction absorbed in the lower right. And then you can look at the ordering of the substituents and see, are there patterns here that would help rationalize why it is that some of these compounds are predicted to have higher fraction absorbed than others. And you can see actually it looks very much to be related to how polar the groups are. So the more polar groups are here, the amino, hydroxyl, etc. So I'll close this, go back here. We can kind of confirm that what we just saw by plotting permeability. This is predicted human jejunal permeability on the x-axis and fraction absorbed on the y-axis. And seeing that the correlation is pretty strong here, as we would have guessed, that those compounds with low predicted permeability are predicted to have lower fraction absorbed. What about solubility? We could plot our predicted solubility at pH 7.4 on the x-axis, predicted human jejunal permeability on the y-axis. I'll take a log scale, and then color by predicted fraction absorbed. And we actually get something like this, where the red compounds or orangish compounds are predicted to have the lower fraction absorbed, whereas the green compounds are predicted to have higher. So you actually see three kind of clusters here. It's actually pretty easy to see what structural elements are in each of the clusters, but I'm not going to have time to do it. I just want to show, if you look at a particular compound here in the cluster of somewhat low permeability and somewhat low solubility, you see this compound here, predicted fraction absorbed is less than 50%. If I select that compound, this is the compound here, I can show the feature, the parameter sensitivity analysis feature, where it's going to look at both solubility and permeability and provide information about which of those is, you can think of it as most responsible for the low predicted fraction absorbed. You get a graph that looks something like this. So the red indicates permeability, the blue solubility. So it's, showing that if you want to try to improve the fraction absorbed of that compound, you need to look at the permeability of that compound, that the solubility has less effect. One interesting thing to note, one last final thing about this compound is that if you were interested in trying to improve the permeability of this compound, that's not always a trivial thing to do without also, drastically lowering the solubility of the compound. For example, you may remove the polar amino group. That would certainly improve the permeability, but one might expect that to also decrease the solubility because you've removed a polar group. But what's interesting, this is just another interesting use of ADMET Predictor that I want to highlight. If I copy this compound to the clipboard and do a substructure search with this compound, but I'm going to modify it by removing that amino group, do an exact search. That compound actually is already in the data set, and it's here. This is actually a commercial drug called glycoside. What's interesting is that it does have a higher predicted permeability, which we'd expect since we removed that amino group. Actually, the predicted solubility at pH 7.4 is actually higher, which is interesting. And it's because of the zwitterionic character of the compound, that the compound with the amino group has more zwitterionic character at pH 7.4, which means that it has less ionization and lower solubility. By removing the amino group, what dominates is the acidic hydrogens on the urea group. So it's more ionized, as it turns out, at pH 7.4. I only show this to illustrate that there are a lot of things you can do with ADMET Predictor. Designing compounds on the fly, actually rationalizing what kinds of structural changes you might make, and then immediately being able to predict properties for those compounds. It's quite a powerful combination of tools. So I walked through that example. It took a little longer than I wanted, but I think it nicely illustrates sort of the almost overwhelming combination of things that you can do with this software. I did not have time to go over the delta models feature, but it's something that we've made very easy to do. So if you evaluate the software, you can try that out on any experimental data that you might have. We've reached the hour mark, so I think at this point I better hand it back over to John, and we can answer any questions that you might have.
Thank you, David. Once again, if you'd like to ask a question using your telephone, please use the hand-raising icon on your control panel, and be sure to enter your unique audio PIN. You may either also type your question using the questions pane on the control panel. Some questions have come in. First one is with respect to slide number 5 in the presentation and the HTPK prediction for both bioavailability and dose required to reach a certain concentration. Can you comment what dosage form and particle size is assumed? And are both fraction absorbed and bioavailability given so the user knows if solubility is contributing to limited bioavailability at the dose model. I think you talked about this a little bit with the new parameter sensitivity analysis feature, but if maybe you can talk a little bit about the HIA files and where users could potentially modify some of those inputs like particle size.
Yeah, I alluded to that briefly. With the, I'm not sure I can actually open it. I'll try to do it while I'm talking, but there is a set of files that has a number of parameters, some of them related to formulation, some of them related to physical chemical properties. I believe there's another set of files that's even related to the overall ACAT model. Okay, I won't even try to find this file, but it's included with the distribution. So yeah, those are things that you can modify if it becomes important for us to expose those parameters within the interface so that it's more convenient for you to modify those things. We would be happy to do that. We've limited the selection of parameters that are actually visible in the interface. We limited those to the ones that we thought early discovery scientists would probably be most familiar with and be most likely to modify. Let's say I may have to ask Bob maybe to comment on some of the other aspects of that question. I think the assumed dosage form, I think it's immediate release tablet. Is that right, Bob?
Yeah, and it's 0.25 micron particle size. Can you hear me?
Yeah, we hear you.
Yeah.
Maybe while the next question's coming in, I'll find that file.
Okay. Thank you both. Question about the PGP substrate and inhibitor models. Can we comment on the cutoff percentage or cutoff values used when making a prediction for either yes or no for substrate and inhibition for the PGP models?
You want me to take that one too, David?
If you would, Bob, please.
Those models are built on published classifications by experts that are actually a combination of several different measurements, including Keiko and calcine uptake measurements. But the main one used for the PGP is an ATPA stimulation assay. And they chose the cutoff, and it was affected by the results in other data, because it's a mix of various published. It's a compilation of published data with varying conditions and things like that. So there is not a set threshold. But what we do is we recommend that people use the confidence attached to the assay essentially as a threshold. That is, if you only accept ones that have a 90+% confidence in the prediction, that's effectively setting a higher or lower threshold depending on how you think about it.
Okay, thank you, Bob. Related to additional models in the PhysChem Biopharm module, do we have predictions inside of ADMET Predictor for blood-brain barrier penetration?
Yes, we do. Bob, do you want to answer that?
We have two models. One is a classification model for penetration itself, and the second is a regression model that predicts the blood-brain concentration ratio. And that actually is controlled by PGP in most cases. So that one aspect is, can it get into the brain? And that's the BBB filter, blood-brain barrier filter. And then the second part is whether it stays there. And that's the log BB model.
Thank you, Bob. David, you seem to have stirred some interest in the design module that's part, or the MedChem Studio module and the design capabilities within it. Can you talk a little bit about the different options for designing structures inside that MedChem Studio module?
Yeah, let's see briefly. I think there are 3 or 4. One is you can sketch an actual reaction scheme using our sketch tool, ChemDraw, AccelerisDraw, and then provide lists of substrates. So for for reactants. So for each of the reactants in your scheme, you can provide lists, explode that. There's a feature that it's similar to one published recently, or within the last 10 years, by Abbott Laboratories. They called it Drug Guru, where there's a database of sort of known either reactions or transformations. And you take a lead compound and sort of iteratively apply that database of transformations or reactions in order to produce analogs. That's a nice way to sort of explore how you might make derivatives of a particular lead structure. And we have our sort of default database of transformations that we provide, and then you can modify that or provide your own. I mentioned this one, or I showed this one. You draw a scaffold, provide a list of substituents, and it explodes those out. And then there is another set that allows you to take a structure and identify a scaffold within that structure, and then you can choose either to explore different replacements of the scaffold using again a database that we provide that's built using known drugs, or you provide your own database. So you can either replace the scaffold or replace the R groups. So it's another way to explore how you might make derivatives of one or more lead compounds.
Thank you, David. Those new fingerprints that were part of the keys feature in MedChem Studio module, can those be used as descriptors in ADMET Modeler?
They can. Yes, they can. Once you generate those descriptors, you can select most likely a subset of those. You essentially convert them into sort of columns in the spreadsheet in the way that our normal atomic descriptors are properties in the spreadsheet. And then when you launch Modeler, those become available for selection.
Thank you, David. Are confidence estimates available for the new multi-class ANNE models?
I believe that they are not. I'll have Bob give the definitive answer, or Marv if he's on the line.
Yeah, no, they're not. The confidence analysis methodology was developed for binary classification only at the moment. It's theoretically possible to extend it, but we haven't done so at this point. It would involve a bit of research. We have some ideas about it, but it's a little tricky. And at any rate, they're not at this point.
Okay, thank you, Marv. For the delta model approach, can that be used on classification models?
Again, I'll have Marv and Bob give the definitive answer, but I believe we do not support that, right?
Yeah, no, the delta models require a quantifiable error difference between the predicted, or between the global prediction model and the observed value. So again, they're not available for classification models. Again, that would involve some theoretical research and methodology development to figure out how to do it for classification models.
Okay, thank you, Marv. For the HTPK simulation module, how does the calculation of the volume of distribution compare with GastroPlus?
Yeah, I kind of had to gloss over the whole issue of volume of distribution. I know I'm going to have to transfer this over to Bob in a second. But you know, you can specify. Let's see. Should probably just hand it over to Bob now. But you know, GastroPlus does provide what we call a mechanistic volume of distribution that involves the individual Kp values of individual tissues. We do calculate that value, and we do provide it as part of the simulation results in ADMET Predictor. And I believe that value just uses the same equation that's used within GastroPlus, and we do provide it. I think when you run the simulation in ADMET Predictor, you can choose whether to use that mechanistic volume of distribution or the predicted volume of distribution from our QSAR model. So I'm pointing to it here. So we have a separate model of volume of distribution that's one of our commercial models, and you can choose whether to use that volume of distribution or the mechanistic one. And I'm sure Bob will want to say a couple of words about this.
It's really, really complicated, but they measure slightly different things in the sense of what the units of liters per kilogram mean. But as David said, you can set the HTPK module in Predictor to use either one. For rats, you're much better off using the mechanistic one. The human volume of distribution model isn't really applicable to rats. The mechanistic one, if you have the other parameters you need, is applicable to any species. But basically, they're measuring slightly different things because one focuses on an equivalent volume of blood, and the other focuses on the equivalent volume of blood that's corrected for how much fat there is in various tissues. They're pretty much equivalent as long as you are down below 1 liter per kilogram. And above that, it's a very abstract notion anyway. So just use what works. You can use either one, as David said, in GastroPlus as well, if you're doing central compartmental analysis and just using ACAT.
Okay, thank you both. There's a request to just briefly describe the various endpoints that are predicted in the toxicity module. David, do you have a slide, or do you mind going to the toxicity tab and just briefly talking a little bit about the different models?
I'm sorry, you guys didn't hear me, but yeah, it's probably best to show it as a schematic. Let's see. So, you know, I'm probably not the best one to be explaining this. The probably least toxicology background of anyone on the phone here, but you know, I did mention the hERG channel blocking, hepatotoxicity measures or predicts likelihood of causing elevation of 5 different liver enzymes. I think I mentioned the endocrine toxicity models. So this is binding to estrogen and androgen receptors. Various carcinogenicity models, main one being 10 separate Ames models. So 5 bacterial strains with and without the metabolic activation. I think I alluded to the sensitization models. I think these are rodent models, I believe. You can correct me if I'm wrong.
No, those are human.
Those are human, okay.
The sensitization.
Bob, you'll have to speak to some of the other ones. MRTD is maximum recommended therapeutic dose.
Yeah, those the one your arrow is close to now. those are all human, except for the last one, which is the acute rat toxicity. The environmental ones are sort of self-explanatory. It's a variety of toxicity to the various aquatic species, how much something is going to break down in pond water, basically. And then the bioconcentration factor is how much stuff accumulates as you move up the food chain. Carcinogenicity is just higher level, more mammalian, more direct measures of carcinogenicity than the Ames test. And endocrine disruption is human receptors.
Okay, thank you both. With respect to the ability to generate metabolites in MedChem Designer, are those specific to a species, or could we apply some of those predicted metabolites, for example, to a rat model, monkey model, dog model, etc.?
I'll just bring that up again.
Well, the sites of metabolism are based on modeling the results of human CYP enzymes. So the metabolites are based on the sites of metabolism models. So in principle, primarily they're intended to work for humans. Those metabolites might be valid for other species to the extent that the CYP enzymes are somewhat similar across species, but they're primarily intended, they're based on modeling sites of metabolism from human CYP data.
Yeah, that's true for the ones shown. We do provide an overall CYP clearance prediction for rat microsomes. In practice, typically the 3A, the CYP3As are represented in most species to a greater or lesser degree, but even that has variations. If we could get the data, we'd be happy to build the models.
1, 3, 1, 4, 5.
We'd be happy to build the models.
Okay. Thank you, guys. And it seems like last question in HTPK simulation module. Is it possible to extend the prediction of the plasma concentration time profile beyond 24 hours? And then a follow-up to that, is it possible to simulate for multiple dose administration?
Not yet.
Okay, so no to both.
No to both. Yep, that's a logical extension to that feature. But for this release, it will be limited to 24 hours, single dose.
Okay. Thank you, David. And I think that is it. So that's going to conclude today's webinar. As a reminder, playback will be available online at our Resource Center. For more information on ADMET Predictor, GastroPlus, or any of our other products or services offered by Simulations Plus, Cognigen, or DILIsym Services, please do visit our website, www.simulations-plus.com. And be sure to connect with us on one of our social media channels for the latest news and events. I would like to again thank David for the excellent and insightful discussions today, and both Bob and Marv for their contributions to the questions. Thank you for joining us, and have a wonderful day. Bye-bye.